Okay, at this point, I'd like to turn us over to our audience questions. A few have come in, and I think they're really good questions. And I will open it up to the floor. Nina, David, feel free to respond. The first one is, given that treatment of children for many diseases will require readministration of the therapeutic, do you see a role for immunomodulatory therapies to prevent immune responses to both AAV and transgene domains?
Yeah, again, that's just where I think it's exciting, the potential for redosing, whether it's mTOR or it's IDZ or other antibody degrading enzymes, or always a new novel approach to sort of induce tolerance or immunosuppress fairly heavily at that given time. So we'll see. Nina's REGENXBIO working, being able to treat the second eye, and others, of course, will starting to be able to do that. Gives us hope, and is depending especially on that with that redosing strategy. Is it still really high dose at 1E14 vg per kg, for example, or can we now redose it at a lower level because we're inducing more of a tolerant response. So I think that the possibilities for a redosing strategy will exist in the future. I'm very hopeful for it, and I think we'll see what happens in the next few years.
Sure. Okay. Next up, are stable producer cell lines capsid type with substitution of cassette on the horizon?
Yeah, sure. I mean, obviously, the Cevic cell line, that is been developed, and others have been working on HEK or CHO-based systems for this. I think that the big question there is, what is the productivity out of those systems? And the nice thing about a producer cell system is that you can apply more process intensification, so perfusion-based systems, and potentially carry the cells longer and get really high productivity, or at least match the productivity we're seeing in transient-based systems. But I'm not aware of anyone right now who's in the clinic using a producer cell, other than, say, the PCL systems that from adenovirus and HeLa from years ago. But true producer cell, I think those are on the horizon. It's just about the time it takes to develop that. Again, even if you have the ad helper functions, herpes virus functions, and have the capsid, it still takes quite a while to develop and clone and make a master cell bank, release that bank to then be able to really start some of your development. And that challenge of time, especially when patients are waiting for drug, is difficult in the rare disease. So what I would see is more likely scenario is people starting with more of the traditional transient transfection-based approaches or baculovirus SF9 approach, and then pivoting to a producer cell either in later phase development or post-approval.
Sure. Okay. And excuse me, David, our second poll question today, we asked our audience members, what do you consider the most significant manufacturing challenges for AAV-based gene therapies? Just interesting to know that 46.2% of our audience today selected reducing empty capsids, followed next by scaling up production, then achieving sufficient viral titers, and finally eliminating host cell contaminants. So just some interesting information. And given that the top, almost half of our audience is really concerned about reducing empty capsids, any color commentary on that poll?
Yeah, I mean, addressed it slightly in the earlier question. Again, I think the tools are better than they used to be, right? The monolith and the resins that we have now to do this through AEX are far superior than what we had 10 years ago, struggling to do this through iodixinol gradients. And I think having better ways to automate, if we're going to stay with the ultracentrifugation based, and make it more automated and not be relying more on that human operator to be trying to pull bands from a gradient, which is difficult to do in a GMP setting. So I think, but the main thing is to be able to really reduce that on the front end. And in the back-of-virus SF9 system, for example, it's achievable. We've been able, in the past, former company, been able to show that you can get 80% full particles coming out of it. So I think it's, and others have presented, depending on the transgene and the capsid, to be able to hit really high full particles at the front end. So you're not having to really enrich. It's more of a polish step when you get to AEX. So I do think it's achievable, but it's probably going to be on a product specific basis to really get that optimization to get that really high full particle. So I do think it's achievable. It's just it's not without its challenges.
Sure, of course. I think we have time for one more question, and that will be, is co-development of CDX becoming a significant cost burden, especially for smaller companies?
I would say yes. I think it's something important to consider, especially just in May when the FDA issued the final rule, aiming to ensure the safety and effectiveness of the LDTs and how this impacts the companion diagnostics that we would want to use for development is something that keep an eye on, sort of the risk-based approach that they put in place. And then also knowing that ex-US, right, there are different requirements. So then how does one think about being able to develop one assay for all of that? And FDA has sort of taken a benefit-risk approach to this, where some sponsors have made sure that they had a companion diagnostic ready for approval at the time of their product approval, and others have taken a more post-market approach to developing the companion diagnostic after initial approval. And so I think it's important to speak to the agency at case-by-case basis, but it is a consideration and cost burden for resources here.